Last lecture was the previous lecture, not the last, the previous lecture was about what happens when the chromosomal number, aneuploidy, polyploidy, what happens, monosomic, trisomic, the chromosomal numbers is altered due to a non-disjunction event during gamete formation. Well, let's change it up a bit. What happens when the chromosomal number is correct, but there's been alteration of the chromosomes themselves? We see here 4 alterations that may occur to individual chromosomes. Deletion, duplication, inversion, translocation. Let's look at this. What's happening here? If you look at the top there, A, the deletion. The gene sequence on the wild type or the normal chromosome, A, B, C, D, E. Due to a deletion, what was A, B, C, D, E in the offspring will now be A, B, C, E. A whole chunk of that chromosome has now disappeared. Where's D? Don't know. Hope it wasn't something important because, well, you now only have 1 copy of it on the other homologous chromosome. Turns out that for some genes, you need to have 2 pairs there, 2 alleles, both expressing to get the correct level of proteins. Was this one of those? Don't know. What about duplication? A, B, C, D, E becomes A, B, C, B, C, D, E. Well, whatever BC is, hope it's not something that has to be kept at a certain threshold level, because since you now have 2 copies on this 1 chromosome, 3 total, because you got to remember you got the homologous chromosome, you're now going to have an increased level of those genes expressed. Again, hope it's not something that has to be kept at a certain threshold. Too much is bad, too little too bad, just right. Because now you're going to have even more of that gene product being made. Could be an issue, especially if this is genes that are required just for early fetal development and have to be at a certain concentration of the gene product. What about inversion? ABCDE becomes ADCBE. Hey, the genes are still there. We're all good, right? No. Try reading instead of left to right, like you've been taught to read in English. Try reading right to left. Pick up any page, any book. Read right to left. The words will not make sense because everything has been spelled out to be read in one direction only. Same with our genes. If things are backwards, they might as well not be there. And then you get what's referred to as the translocation, the reciprocal translocation. AB, which was on chromosome 7, swaps with MNO from chromosome 12. The genes are still there, they're just now different loci. Will that matter? For this, maybe, maybe not. Changing of the genes could change whether or not the transcription proteins, the proteins that are there for expression of genes, can be able to locate and bind. And if it's not going to be an issue for this individual, what about for their offspring? Chromosome 7 that did have AB, yes, there is another chromosome 7 that has AB there. But hey, remember during gamete formation, boom, going to end up with half the gametes that have MNO on chromosome 7. No AB. Ruh-roh, Shaggy. And then their 12, which should have had MNO, 50-50 chance it's going to get the correct one, incorrect one. So AB could completely disappear. Their offspring would be the deletion. Some of their offspring could be a different form of that duplication. Too many MNOs, no ABs. Too many ABs, not enough, no MNOs. Weird things happen. So let's look at some human disorders that are due to some form of chromosomal alteration, altered number, or some change in the chromosome structure itself, the sequence. Like I just stated in the last slide, the sequence may not be, if it was an alteration, it may not be during the fetal development. It could have been something that occurred in the parents, but it was masked because there was the homologous chromosome there that was able to at least give a phenotype. And then it's passed in the gametes to the offspring, which now that's going to be seen even more so. Trisomy 21, Down syndrome. Trisomy, okay, means three chromosome 21s. Okay, 1 in 700 here in the United States. Going to stand on my soapbox here. If you like it, great. If not, oh well, it's my class, my soapbox. Turns out the U.S. has one of the higher in the civilized industrial world, first world, however you want to phrase it. 1 in 700. Some people want to sit there and say that's because of our environment here, our bad prenatal health care, blah, blah, blah. And they point to Europe and say, look, they're also first world, civilized world, whatever you want to call it. And their rates are much, much lower. Straw man. It's not the same thing. It's comparing apples into oranges. And the reasonings their numbers are so much lower has absolutely nothing to do with environment. It has nothing to do with healthcare. It's turns out one of the leading causes for abortion in many of the Scandinavian countries is a positive fetal test for trisomy 21. Turns out they're just aborting all of the Down syndrome babies. Whereas here, since we are, pro-life, pro-choice mix, we're not quite so pro-choice as they are, we have much higher rate. And when you look, it's right here. 3 chromosome 21s. Something about chromosome 21 calls for an altered gene expression during fetal development, gives the altered craniofacial appearance, also has an effect on mental acuity, mental development, physical development. It's why many people who have Down syndrome are going to have a very shortened lifespan. They're going to have there's a wide range of mental abilities. Some are going to be high functioning, basically have the mentality 12, 13-year-old. That's about as advanced as they can get, mental acuity wise. Some are going to be stuck, no matter what their age, 2, 3-year-old mental acuity. Physically, shortened arms. They're going to have that distinct body shape that you see. But again, it's going to be a spectrum. One thing that's weird that they see predominant in most people with Down syndrome, they are happy, non-stressed. I mean, they can be upset, they can be sad, they can get stressed, but they're happier than we are. Lower quotient for lying. They just don't seem to have, many of them just don't seem to have it in them for deceit. Go figure. Don't know. Then you also get the sex chromosomes, non-disjunction events in the sex chromosomes. Um, yes, you do have the true people who are hermaphroditic. You do have people who are somewhere on a spectrum of development, not sexuality, development. You get Klinefelter syndrome, um, XXY. Because of the Y, the SRY gene there, they will have the gonad material will still drop through the abdominal floor, will develop as testes, but because they have the XX, the double X there, lots and lots of estrogen, these are going to be males who are going to be more feminine in appearance. Okay, not all effeminate men are Klinefelter. The ones that are Klinefelter, um, you will see jaw, cheekbones, their skeletal structure is definitely skewed way towards a more feminine. The physical skeletal markers and things that we would associate with female versus male, because of the double X, Klinefelters, it will be there. The wider pelvis, things like that. We also see a spectrum of different things that occur in females. Um, one of them I don't see here that I may have forgot to put on the slide, XX partial Y. XX, female. The partial Y means that it's part of, not the whole Y, just part of got stuck during gamete formation for the sperm. So their SRY gene is not present, but some of the other genes present for male development is. So for these XX partial Y, these women will be, I hate to say it, but please don't be offended. These are going to be females that are very mannish in appearance. Okay. Skeletal structures are going to have markers that are more male. Bone structure, musculature is going to lean very heavy male. Not to say that all women that are broader, bigger, um, are these XX partial Y. It's just that these women that are XX partial Y will definitely skew more towards this male-ish appearance, heavier, facial hairs, things like that. You also end up with Turner syndrome, XO, monosomy X. This means that either the sperm or the egg was not carrying a sex chromosome. These women will be sterile. Okay, turns out you do have to have both X's there to be for sexual maturation, both X's there for the normal cycle and all that to occur. When you go and you start looking at further, alterations to the chromosomes, instead of just numbers, presence, absence, as we saw in the last couple of slides, what happens when you alter the structure of the chromosome itself? If you were to go through and delete portions of chromosome 5, you're going to end up with a developmental disorder referred to as Crudeshot. Sorry, my French is not that great. Basically, cry the cat. These individuals, physical, mental, extreme retardation. Physical, mental. Brain does not fully develop. Skeletal muscles do not develop. These will be young children. They do not make it generally past infancy, childhood, into adolescence. The name comes from the fact that that's all they can do. Cry of the cat. Sounds like a cat caterwauling. There is no higher brain function present. Other disorders, certain types of leukemia, are due to translocations of chromosomal parts. Genes are still there. You go through and you test for the presence of the genes. Ooh, yep, they're there. Come up positive. Everything's great. Until you go and you sequence the chromosomes that they happen to be on, and they're in the wrong place. Wrong place, wrong address means that the gene expression information that we're going to be talking about 2 chapters from now can't locate those genes. Then you're going to get things that just don't fit. More exceptions to Mendelian genetics. I'll let you read on your own genomic imprinting. Basically, in a nutshell, environmental cues to the cells will tell the cells, especially the ones that are forming gametes or during field development, to mark one chromosome over the other using either methylation or some form of other molecule marker, and basically that says choose this chromosome over that one when you go to express a gene. I will put these are on the chapter page in Canvas. 9 things, or actually 8, don't really so much worry about the genetic imprinting. 8 things you need to know, be able to discuss for this chapter.